All right, let's talk a little bit about pulmonary ventilation and get into some math, which I know is your favorite. But what's happening here is that inspiration and expiration that we talked about just a few minutes when I went over that pulmonary ventilation box with you. The inspiration and expiration, those, like I said earlier, are going to occur to differences in air pressure. And you need to understand how this works. There's another great animation here on Boyle's Law. Boyle's Law is the math of how this works, the physics of how this works. What's happening here is, there we go. Let's look here. So you've got inspiration on one side and expiration on the other, and you've got your volume of your thoracic cavity shown in the top boxes, and you can see it's smaller over here and larger over here, and you can look at the changes that are taking place because of your diaphragm pulling down, your diaphragm pushing up, and how that's related to the expansion of the thoracic cavity or the constriction or closing in of the thoracic cavity. It also causes the vertical changes. Well, I guess that's really just talking about vertical changes of the diaphragm. You have lateral changes in your thoracic cavity as well, so these changes here, and that's highly due to your ribs. And so your ribs being elevated to widen your thoracic cavity like you see in inspiration, or the ribs kind of depressing and squeezing in to constrict and compact that thoracic cavity on expiration. And then you also have your sternum moving out or moving in to, again, contribute to the size differences of the volume on the thoracic cavity. So this is volume on this picture, and that's part of Boyle's Law. Then the other part has to do with pressure. So what you've got here is, well, let's walk through this diagram here first. These are like kind of like little pistons in cylinders, if you can imagine that. And you've got a certain amount of space with a certain amount of molecules here. But if you were to push down on this piston, it would squeeze things, and that's going to have the same amount of molecules, but now you're in less space. So that's a change in volume, and it's a change in pressure. So the rule here is that pressure is going to decrease as the volume increases. So this is a bigger volume, and we have decreased pressure. Flip side of that, pressure increases, increased pressure, as volume decreases. So when we squeezed it and made that space smaller, it's going to decrease the volume, and that in turn increases the pressure. That's Boyle's Law, that relationship. So the gas pressure is going to decrease as the container volume increases. So we call this an inverse relationship. We've talked about some of these before in the past. That just means that as one thing goes up, the other thing goes down. And our two things here are the pressure and the volume, which we're going to abbreviate as V. The second part of this picture here, part B, is showing how the air is going to move down the pressure gradient. And it's kind of like diffusion, but we're using a pressure gradient instead of a concentration gradient because we're talking about air instead of talking about solutes. If you are moving down a pressure gradient, you're moving from the highest pressure to the lowest pressure. So it's similar to that pattern that we see in diffusion with solutes moving. But it's essentially the diffusion of the gas down the pressure gradient. So what happens here, there is no difference here, so you wouldn't have any net movement. Yes, you could have some molecules moving that direction, but you'd have them moving this direction at the same rate. Here you've got your decreased pressure because you're at a bigger volume, right? Just like we talked about over here. So here we're going to go from an increased pressure, high pressure, to low pressure. So it's moving into this part of the container. And the opposite over here. This is high pressure here, low pressure here. Again, because you've got less volume here and more volume there, same amount of dots. So you're going to go up in that situation. So you go from high pressure to low pressure. That is the way the air is going to move. Here's your math, and you do need to know how to do this math. P1V1 equals P2V2. If you haven't had an algebra class in a while, what this means is that pressure 1, the pressure in one place, times the volume in that first place, and you multiply those together. And that's going to always equal the pressure in the second place times the volume in the second place. So it's not a squared or anything like that. The ones and twos are just to denote which region are you in. And we're going to see different body regions as we move on. So here's some math to just try it out before we talk about this stuff here with the different pressures in the different parts of your body. Do this equation, do this problem. So P1 is 15, V1 is 10, P2 is 30, so what is V2? How would you do this problem? Pause the video and try it out. You should get 5 as your answer. And if you didn't, let's go through this. Hold on one sec. All right, so if P1, you have to remember that formula. P1V1 equals P2V2. And the way you're going to do this is some algebra. You are trying to solve for V2, so you divide both sides by P2. That makes that and that cancel out. So now you're left with P1 times V1 divided by P2 to find out your V2. And then you just plug your numbers in. So it says P1 is 15, so we take 15. Remember, the two next to each other just means that you're going to multiply. P1 times V1, V1 is 10. And then P2 is 30. And that's going to be equal to V2. Then you just do your math. 15 times 10 is 150. 150 divided by 30 is V2, and you can cross out your zeros. 15 divided by 3 is 5. So that's how you should get the 5. All right, so now let's look at a little more depth about these different spaces and pressures that we're going to be talking about. So there's going to be atmospheric pressure. Atmospheric pressure is the pressure of the air that's in the atmosphere, that's outside of your body. So that's like out here. The intrapulmonary pressure is the pressure that's going to be inside the alveoli. So intra is within, the pulmonary meaning the alveoli. So that's in these air pockets called the alveoli. That's the pressure inside of there. And then because you have that pleural cavity between the two parietal and visceral layers of the pleura, you're going to have pressure in there too. And that's going to be the intrapleural pressure. And this, the IP pressure, is always lower than the intrapulmonary, this one. So this, you could say, is always higher than that, or this is lower than that, however you want to think about that. And the reason for that is you want your lungs to stay inflated like a balloon. You don't want them to collapse. And if you've ever heard of a pneumothorax, that's a problem with all this pressure. They talk about that in a clinical view in your book as well. So you've got to have higher pressure in the alveoli than you do in that pleural cavity so that your lungs are puffed up like a balloon. All right, so now we are to this crazy diagram. Here we're looking at quiet breathing, and you're looking on the left at quiet inspiration and on the right at quiet expiration. You can see how the volume changes in your lungs, and you can see how the pressure changes in your lungs. And we're going to go through step 1, step 2 for inspiration, and then step 3 and step 4 for expiration. And quiet breathing, you can read more about this in your book, but quiet breathing is the breathing that you don't really think about. It's just kind of autonomically happening. It's not what we call forced breathing. So that's forceful breathing, whereas, I've been breathing this whole time, and hopefully you haven't been able to hear me breathing. So that's quiet breathing. All right, so let's look at these. Let's look at the inspiration side first, and you've also got this awesome summary chart as table 23.3. So the first thing is the inspiration. During quiet breathing, your diaphragm is going to contract and your external intercostal muscles are going to contract. So you see that here in the summary chart. Another good one to copy and keep as a reference point. And the reason that you're doing that, like I showed you in those diagrams earlier with the box, the purple box and the green box, that's going to expand your thoracic cavity and allow it to be the bigger volume. You are going to have atmospheric pressure at a certain amount of mercury. That's how like barometric pressure is measured. So, that fluctuates all the time with seasons and weather and things like that. But this is just going to be our, for instance, barometric pressure for this example. So about 760 millimeters of mercury. The intrapulmonary pressure, which that's the pressure that's in the alveoli, which hopefully you remember, that is 760 at this moment. And then what's going to happen when we expand the thoracic cavity, we're going to give it a bigger volume. When we give it a bigger volume, what's going to happen to this pressure here? It will lower. If you look here, it's lowered by 1 millimeter, but that's enough to make a difference. So when you get that lower pressure due to the larger volume because you've expanded your chest cavity, that is then going to make the air flow in. So that's why you see the air arrows, the black arrows flowing in, and it's about 500 milliliters or half a liter for every quiet breath. That of course can vary depending on a person to person, but you're going to go from that high atmospheric pressure to the lower intrapulmonary pressure now. So that's why air goes in. You don't have to force it in. It's just going to automatically go in your lungs because of the pressure changes. But the pressure changes won't happen unless your muscles contract. So down here again, we see pleural cavity volume increases, pressure decreases, and the lungs, same thing. And then down at the bottom, air movement into the lungs. So this chart is your awesome summary. All right, looking at the expiration side of things, expiration is also going to happen just passively, but again, because the pressure is going to be different. So here we've got the muscles are going to relax, and when the muscles relax, that's going to allow the volume to decrease. So that's the other side of that purple and green picture that I was showing you earlier. So the volume's going to decrease now. So if volume decreases, remember Boyle's law, pressure increases. If pressure increases, down here, now we've got high pressure in the lungs compared to the lower pressure to what, and it's the same, it's just low now compared to this higher pressure. Low atmospheric pressure. And so that's going to push air out of your lungs. And again, it's just a quiet breath. It's a passive movement. It's not really a push. It just, the air is going to go from that high pressure region inside of your lungs to the low pressure region. So it's everything is the opposite in this chart. So do you have to memorize two sides of it? No, just memorize one and then know that everything is the opposite. So you've relaxed those muscles, your volume decreases, so your pressure increases, and then the air is moving out of your lungs. So that's quiet breathing. Forced breathing. They've got a picture here showing you some muscles that you can also contract and relax to do your forced breathing. So this is like when you tell yourself, take a deep breath, and you really do those forceful inhales and exhales. What's happening here is you've got all these additional muscles that are contracting or relaxing, and so you can get bigger volume changes. And when you get bigger changes in volume, you will get bigger changes in pressure. Bigger changes in pressure mean bigger changes in the amount of air coming in and the amount of air going out. And so it's going to make more noise and it's a more forceful breath. So some of these we learned in 210, sternocleidomastoid, your internal intercostals, external obliques, transversus abdominis, and external intercostals and diaphragm were the ones that we use during quiet breathing. So these are the ones, you don't need to know their names. I was just pointing those out to give you some references, making some connections with 210. All right. Nervous control. So lots of different things here with nervous control and how you can regulate and control your respiratory rate. You've got a structure called your respiratory center. You actually have two. Those are going to be centers in the brainstem. So you've got the medullary respiratory center, and that's going to be in the medulla oblongata. You should have learned a little bit about these in chapter 13 in BIO211, but this is what's going to regulate your respiratory rate and the depth of your breathing, so deep breaths versus shallow breaths. It actually is broken down into two sets of nuclei. So the VRG, or ventral respiratory group, and then the DRG, which is the dorsal respiratory group. And if you've forgotten nuclei when we're in the central nervous system, that just means clusters of cell bodies, if y'all don't remember that term. So cell bodies of neurons. And we're in the medulla oblongata, and there's a dorsal and a ventral. And one's dorsal is the same as posterior, and anterior is the same as ventral. So the VRG, or the anterior group, is what sets your normal quiet rate, and the DRG is what's going to change that rate and depth based on information coming in from chemoreceptors. And you see some blue arrows coming into this diagram. And so when we expand this, you can see chemoreceptors coming from all over the body. Like this. So chemoreceptors coming from all over the place, and lots of things causing those respiratory centers to respond. So you also have a pontine respiratory center, and that's up here in the pons. That's where you regulate the rhythm of your breathing. And then down here, you also see that you've got motor signals carried For inspiration, those are going to be carried by your phrenic nerve. Your phrenic nerve is one of those cranial nerves that we learned also back in, actually it's not a cranial nerve, I was thinking of the vagus. Your phrenic nerve is a nerve that we did learn in 210 as part of one of your spinal nerves. So that's, the phrenic nerve is a really, really important spinal nerve that innervates your diaphragm. And it's branching off of your cervical plexus. I think we learned that in 210 also. So phrenic, I remember that, just a little trick. Phrenic has a pH and so does diaphragm. So it's a little trick for learning that. And you also have intercostal nerves, if you remember that from 210. Inter means between, and costal means ribs. So these are thoracic spinal nerves, and they run between your ribs. So you see them there. Those nerves are going to go and innervate the external intercostals. So that's how you tell your diaphragm and your intercostals to relax and contract depending on what's going on. So during inspiration, if we go back to that little chart, remember your diaphragm and your external intercostals are contracting. So that's what's going on here. The phrenic nerve and the intercostal nerves are telling these two sets of muscles to contract during inspiration. And chemoreceptors are, monitoring things. So these are receptors, sensory receptors, like we learned about in chapter 16, and they are monitoring the presence or absence of certain chemicals. They're very, very specific, monitoring different things. Here you've got chemoreceptors for carbon dioxide, hydrogen ions, and oxygen. They're monitoring your blood. They're also monitoring your CSF, your cerebrospinal fluid. And the most important one to monitor there is the CO2 levels. CO2 can be toxic if it's in high values. So that's really, really important to monitor that so that you can regulate and change things as needed. But you see these chemical receptors or chemoreceptors up here in the top box. We will talk more about exactly what happens when the CO2 gets too high in the last video. Let's see. It is a reflex, so you're going to adjust your breathing rate and the depth of your breathing as needed to maintain airflow. And again, that airflow is going to depend on pressure gradients, and it also has to do with resistance in the air passages. So bronchodilation and bronchoconstriction come into play, but we'll get into that. I think that's in the last video as well. Yes. So more on that coming up a little bit later. So wrapping up here for this video, talking about the lungs, is looking at the respiratory volumes. And we do cover this in the lab, but you may not have learned this in lab yet. So I'm going to quickly go over it real quick in this chapter. But it is explained in your lab manual as well. So you're looking here at lung volume in milliliters. And this is going to show you lots of different things here. You've got your normal tidal volume, which is the normal up, down, up, down, up, down in the middle of the curve here. But you have something called the inspiratory reserve volume. So when you inhale really big, your volume can increase. So this is like your quiet breathing and then your forced inhale. So once you add the two of those together, this is your inspiratory capacity. And that means like your capacity is the ability to do something. So that's how much ability you have to inspire or bring air into your lungs. You've got kind of the same thing over here on the expiratory side of things. So you've got your tidal volume going up and down. And then you have an expiratory reserve volume. So that's pushing extra air out. And when you, I guess we don't call that the expiratory capacity. Anyway, when you push that extra air out, there's going to be a little extra. I call this the leftover volume. It's called the residual volume. That is going to be around 1,000 milliliters of air that stays in your lungs no matter what. You cannot exhale all the air in your lungs. You just can't do it. You would die. You've got to keep some air in there. So that's this residual volume. So that is combined with that expiratory reserve volume. That's your functional residual capacity. So beyond a normal breath, what can you bring into your lungs and what can you push out of your lungs and what's got to be left in the lungs. And your book has better definitions of this. Your vital capacity, look where it is, all the way as high as you go, so as much air as you can bring in, all the way down to as much air as you can push out. That's your vital capacity. And total lung capacity would be absolutely everything, including that residual volume. So you can do a lot of math here, and you've got a chart in your book to explain these. You're going to see things like IRV and TV, ERV, RV, and VC, and then I think they do abbreviate this as TLC, total lung capacity, because we kind of just went over that. The IRV and the TV is what inspiratory capacity is. The ERV and the RV is functional residual capacity. You could say ERV plus inspiratory capacity or plus IRV and TV is your vital capacity. And if you added everything up, that's your total lung capacity. So we will do some practice with this in the lab. But like I said, at that middle pink bar is your quiet breathing, where you're inhaling and exhaling, or inspiring and expiring the air in and out. Forced breathing, going air in, is the inspiratory reserve volume. And then forced breathing, pushing air out, is the expiratory reserve volume. Air left in the lungs after a forced expiration is the residual volume. And then I just kind of took those and put them in little text boxes so that you can match them up. So pause it and see if you can match them up with the definitions I just gave you. And go back to the last slide or rewind the video to check your answers. Or there they are. And there's the chart in your lecture book, showing you the volumes and the capacities with definitions and formulas. We're not going to do these formulas in lecture, but we will do the math in lab. All right, one more video.